In this video, we are starting a very important unit, which is cell biology, specially focused on GAT-B exam. This topic is very scoring because many direct as well as conceptual questions come from here every year. In this lecture, we will cover cell structure, cell cycle, and cell signaling step by step in a very easy and clear manner so that you can understand and remember properly. Now, as you can see on the slide, this lecture is part of your GAT-B preparation and it is designed in such a way that even if your basics are not strong, you will still be able to follow everything. So just focused, and by the end of this lecture, your concepts will become much stronger and exam oriented. Why BioBrains? In the slide, it is explaining why this course or platform is useful for your preparation. Basically, the focus is on concept-based learning, which means instead of mugging up facts, we will understand the logic behind every topic. This is very important for GAT-B because nowadays questions are more application-based, not just direct theory. Also, it mentions that you will get important MCQs, previous year questions, mock test, and proper guidance from registration to counseling. This means your preparation will be complete and structured. So, you should treat this lecture not just as theory, but also a base for solving questions and improving your exam performance. As given in the slide, this lecture will cover structural, functional, and regulatory aspects of cells. That means, first we will understand how the cell looks like, then how it works, and finally how it is controlled. This combination is very important because GAT-B questions often connect these three areas together. Also, the lecture focuses on frequently asked concepts, which means we are not just wasting time on unnecessary topics. Everything we study here has high probability of coming in exam. Along with that, molecular mechanisms are also included. So, you will understand not just what's happened, but also how it happens inside the cell. Prokaryotic versus eukaryotic cell structure. Now, we are starting with one of the most important topics, comparison between prokaryotic and eukaryotic cells. This is a very common question area in GAT-B, sometimes direct, sometimes conceptual. So, you must be very clear about differences and features. Basically, all living organisms are made up of cells, but these cells are of two main types, prokaryotic and eukaryotic. Prokaryotic cells are simple and smaller, while eukaryotic cells are more complex and organized. Understanding this difference will help you in microbiology, genetics, and even cell signaling topics later. Classification of cells. According to this slide, cells are broadly classified into prokaryotic cells and eukaryotic cells. Prokaryotic cells include bacteria, while eukaryotic cells include animals, plants, fungi. So basically, most complex organisms like humans belong to eukaryotic category. The classification is based on three main factors: presence of a true nucleus, level of cellular organization, and presence of membrane-bound organelles. This is very important because if nucleus is absent, it is prokaryotic, and if present, it is eukaryotic. The simple logic can help you solve many MCQs quickly in exam. General features of prokaryotic cells. Now, let us understand prokaryotic cells in detail. These cells are very small in size, around 0.5 to 5 micrometers, and they are structurally simple. Their genetic material is present as a single circular DNA, which is not enclosed inside a nucleus but present in a region called nucleoid. Another important point is that prokaryotic cells do not have membrane-bound organelles like mitochondria or Golgi body. They have 70S ribosomes, which are smaller but work faster in protein synthesis. Also, they have a cell envelope, which includes plasma membrane and cell wall made up of peptidoglycan in bacteria, which provides protection and shape. Specialized structures in prokaryotes. Apart from basic structure, prokaryotic cells also have some specialized structures will help them survive better. For example, flagella are used for movement, and this movement is powered by proton gradient. So bacteria can move towards favorable conditions. They will have pili or fimbriae, which help in attachment and also in conjugation, which is a method of genetic transfer. Capsule is another important structure which protects bacteria from host immune system. Also, plasmids are extra DNA molecules that often carry antibiotic resistant gene, which is very important in medical and exam oriented content. General features of eukaryotic cells. Now coming to eukaryotic cells, these are larger in size, around 10 to 100 micrometers, and much more complex than prokaryotic cells. The most important feature is the presence of true nucleus, which is surrounded by a nuclear membrane and contains linear DNA. In addition to that, eukaryotic cells have membrane-bound organelles like mitochondria, endoplasmic reticulum, Golgi, etc., which divide the cell into compartments. Their ribosomes are 80S, which are larger compared to prokaryotes. This compartmentalization also better control and efficiency of cellular processes. Major organelles and functions. This slide explains different organelles and their functions, which is very important for both theory and MCQs. The nucleus stores DNA and controls transcription, while mitochondria are responsible for ATP production through oxidative phosphorylation, also known as powerhouse of the cell. Endoplasmic reticulum has two types, rough ER for protein synthesis and smooth ER for lipid synthesis. So each organelle has a specific role and you should clearly remember these functions. Now, this slide gives a direct comparison between prokaryotic and eukaryotic cells, which is very important for revision. Prokaryotic cells do not have nucleus, while eukaryotic cells have a proper nucleus. Similarly, DNA is circular in prokaryotes and linear in eukaryotes. Also, prokaryotes lack organelles, while eukaryotes have them. Ribosomes are 70S in prokaryotes and 80S in eukaryotes. Size is also different, with prokaryotes being smaller and eukaryotes larger. These differences are very frequently asked in exams, so you must revise this table multiple times. Cell cycle and cell growth control. So now we are moving to the next very important section, which is cell cycle and cell growth control. Till now we studied about structure of cells, but now we will understand how cells grow, divide and regulate themselves. This topic is extremely important because many questions in GAT-B are directly based on cell cycle phases and regulation. Cell cycle is basically the life cycle of a cell from one division to the next division. But it is not just random division. It is a highly controlled and regulated process. If this control is lost, it can lead to serious problems like cancer. So, understanding this topic is very important not only for exam but also for real biological understanding. Overview of cell cycle. As shown in the slide, cell cycle is defined as an ordered sequence of events that lead to cell division. It is not a single step, but a series of well-organized stages that ensure proper duplication and division of cells. These are four main phases. G1 phase where the cell grows. S phase where DNA replication happens. G2 phase where the cell prepares for division. And M phase which includes mitosis and cytokinins. You should remember the sequence very clearly because many MCQs are based on an order and function of these phases. Cell cycle phases in detail. Now let us understand each phases in more detail. In G1 phase, the cell increases in size and synthesizes proteins required for DNA replication. This phase is also very important because the cell decides whether to divide or not. So it acts like a decision point. In S phase, DNA replication takes place, meaning each chromosome is duplicated. Then comes G2 phase, where the cell checks for errors in DNA and repairs them if needed. Finally, in M phase, the cell divides into two daughter cells. So overall, the cycle ensures accurate duplication and distribution of genetic material. Cell cycle checkpoints. Now, very important concept, checkpoints. These are control points in the cell cycle that ensure everything is going correctly. If any error is detected, the cycle can stop until the problem is fixed. There are three main checkpoints. G1S checkpoint checks for DNA damage before replication. G2M checkpoints ensure DNA replication is complete, and metaphase checkpoint ensure chromosomes are properly aligned before separation. These checkpoints are like quality control systems, preventing errors from passing to daughter cells. Cyclins and CDKs. Now we come to one of the most important molecular concepts, cyclins and CDKs. Cyclins are regulatory proteins while CDKs are enzymes that become active only when bound to cyclins. Together they form cyclin CDK complexes. These complexes control the transition between different phases of the cell cycle, especially G1 to S and G2 to M transition. The key point is the cycling levels change during the cycle, while CDKs remain constant. This regulation ensures that cell cycle progresses in a controlled manner. Tumor suppressors and growth control. Now let us talk about growth control proteins, especially tumor suppressors. These proteins prevent uncontrolled cell division. Two very important ones are P53 and RB protein. And questions from these are very common in exams. P53 is known as the guardian of the genome because It detects DNA damage and either stops the cell cycle for repairing or induces apoptosis if damage is severe. RB protein controls the transition from G1 to S phase. If these proteins are not working properly, cells can divide uncontrollably, leading to cancer. Apoptosis, programmed cell death. Apoptosis is a very important biological process which means programmed cell death. Unlike accidental cell death, this is a very controlled and regulated mechanism that removes unwanted or damaged cells from the body. This process involves enzymes called capsases which break down cellular components in a systematic way. One key feature of apoptosis is that it does not cause inflammation because the cell content are neatly packed and removed. This is important for maintaining tissue health and preventing diseases. Now we are entering the third major part of this lecture, which is cell communication and signal transduction. This topic explains how cells talk to each other and respond to signals, which is essential for coordination in multicellular organisms. Without communication, cells would not be able to function properly as a system. For example, hormones, growth factors, and neurotransmitters all depend on signaling pathways. This topic is highly important in GAT-B, especially pathways like MAPK and receptor mechanisms. As shown in the slide, cells communicate using chemical signals called ligands, which binds to specific receptors present on or inside the cell. This binding triggers a response inside the cell. There are different types of communication. Autocrine, where the cell signals itself. Paracrine, where nearby cells are targeted. Endocrine, where signals travel long distance through blood. Understanding these types is important for both theory and application-based questions. Now, let's briefly understand each type of signaling. In autocrine signaling, the same cell releases and responds to the signal. In paracrine signaling, nearby cells are affected, so signals act locally. In endocrine signaling, hormones travel through bloodstream to distant target cells, making it long-distance communication. These differences are based mainly on distance and method of signal delivery, which is often asked in the exams. Now we will understand receptors, which are very important in cell signaling. Receptors are proteins that receive signals from outside the cell and convert them into a response inside the cell. Without receptors, even if signals are present, the cell will not be able to respond. There are two types of receptors: cell surface receptors and intracellular receptors. Cell surface receptors include G protein coupled receptors and receptor tyrosine kinases, which bind to ligands that cannot cross the membrane. Intracellular receptors like steroid hormones receptor are present inside the cell and bind to lipid-soluble molecules that can easily pass through the membrane. Now let us understand how a signal actually produces a response inside the cell. This process is called signal transduction pathway. It starts when a ligand binds to a receptor which activates the receptor. After receptor activation, the cell is amplified through multiple steps. meaning one signal can produce many molecules inside the cell. Finally, this leads to a cellular response such as gene expression, enzyme activation, or cell division. This stepwise process ensures that signals are properly transmitted and regulated. Second messengers are small molecules inside the cell that help in transmitting signals further after receptor activation. They play a very important role in amplifying the signal and spreading it quickly inside the cell. Some common examples are CAMP, calcium ions, Ca2+, and IP3. These molecules act as intermediates between receptors activation and final cellular response. Because of second messengers, even a small signal can lead to a strong and rapid response inside the cell. Now, we are discussing one of the most important signaling pathways for GAT-B, which is MAPK pathway. This pathway is commonly asked in exams, so you must understand the sequence properly. In this pathway, a ligand binds to a receptor, tyrosine kinase, which activates RAS protein. Then a cascade of proteins is activated in sequence: RAF, then MEK, and then ERK. Finally, ERK enters the nucleus and changes gene expression. This pathway is very important for cell growth and differentiation, and its mutation can lead to cancer. After a signal has done its job, it must be stopped. Otherwise, cell will remain continuously active, which can be harmful. This process is called signal termination, and it ensures that signaling is temporary and controlled. There are different mechanisms for this, such as degradation of the ligand, inactivation of the receptor, and dephosphorylation of signaling proteins. These steps help in turning off the signal and bridging the cell back to normal state. Integration and exam focus. This slide is very important for revision and exam preparation. It shows how all topics are interconnected. Cell structure determines function, cell cycle controls growth, and signaling regulates all these processes. For GAT-B exam, you should focus on key areas like cyclins and CDKs, tumor suppressors like P53 and RB, MAPK signaling pathway, and differences between prokaryotic and eukaryotic cells. These are high-yield topics and are frequently asked in different forms. This slide is more about staying updated and continuing your preparation journey. It reminds you keep learning and stay connected with the resources that provide regular updates and guidance for GAT-B preparation. Consistent preparation is the key to success, even if you study small topics daily, but with clarity. You will build strong concepts over time. So always stay disciplined and focus on your goals.